Let's recap what we know about the cell so far. So this is what you should have learned about the cell in your undergrad degree. So let's go down memory lane. Okay, firstly, we are going to look at the structure of a plant and an animal cell as seen under a light microscope. We will then move on to look at the structure of an animal cell as seen under the electron microscope, and then the structure of a plant cell as seen under the electron microscope. Then we will compare the differences between a plant and an animal cell, and then we will look at the two types of cells, a prokaryotic and a eukaryotic cell. What are the differences between these two different types of cells? And then we will look at the structure of a prokaryotic cell, and then we will look at the difference between a prokaryotic cell and a eukaryotic cell. So let's take that trip down memory lane. Okay, welcome back to the second part of this video that looks at walking down memory lane to look at the structure of plant and animal cells. So welcome back to the honor students for the Life Science for Educators course. Okay, in the second part of this video, we're going to look specifically at the structure of both a plant and an animal cell, and then you're going to get to compare the structure or So we're first going to look at the detail that can be seen under the electron microscope, and then you will be asked to go and look into the textbook and find the functions of the different organelles or structures that are indicated, and just provide a brief description of how these structures are suited to their function. Okay, so let's look at the structure of an animal and plant cell as seen under the electron microscope. Now, there is an animal cell. This is actually a human lymphocyte, white blood cell, and it's seen under the transmission electron microscope. So note the detail. You can see the nucleus quite clearly. The mitochondria are clear. You can also see small vacuoles, Golgi apparatus, the endoplasmic reticulum. So quite a lot of detail can be seen. Now this is a plant cell, and again there you can see the nucleus. The vacuole is clearly seen. Now please note it's segmented here, because remember this is a cross section of a plant cell, and you can see the cell wall quite clearly, and that's how you know that this is a plant cell. Here is another plant cell. Now in this plant cell you can also see the chloroplast there with their double membrane, and the thylakoids where the chloroplast, or should I say chlorophyll, is found. Again, you can see the cell wall, and the cell membrane is on the inside of the cell wall. Now, here is a plant and animal cell side by side. Please note how the endoplasmic reticulum is clearly visible. You can see the mitochondria in the animal cell. You can see the ribosomes on the endoplasmic reticulum, so this makes it rough endoplasmic reticulum. There in the plant cell you can see the cell wall, you can see the chloroplast again, the large vacuole, you can see mitochondria in the cell, so you can see quite a lot of detail under the electron microscopes. Now, this is an electron microscope. This will be dealt with more detail in the next session, but that is the transmission electron microscope, where you have to make sections through whatever you're viewing, and that is the kind of image you will get from the transmission electron microscope. Here is a scanning electron microscope. It's very different in structure because they work in different ways, but those are the kinds of images you can see from the scanning electron microscope, where you get to see the whole structure. Look, that is blood cells, so animal cells there. So you can see you can get different images from both the transmission electron microscope and the scanning electron microscope. Now here are some of the organelles in more detail. So there you can see the Golgi apparatus with vesicles, chloroplast, you can clearly see the double membrane and the thylakoids there in the mitochondria. Those black sections there in the mitochondria are probably the mitochondrial DNA. You can see the endoplasmic reticulum and the cluster of ribosomes. And these would be taken with the transmission electron microscope. Now, here is a nucleus as seen through an electron micrograph from the transmission electron microscope. You can see the endoplasmic reticulum, and it's found throughout the cytoplasm. Here is a scanning electron micrograph of a nucleus where you can see the nuclear pores. Here you can see the lysosomes and the peroxisomes, the mitochondria, the rough endoplasmic reticulum. So a lot of detail is seen, and you can see the scale there is 1 micrometer. So there is a plant cell, a drawing and showing the ultra structure with all the organelles that are in the cell. And here is an animal cell. So note the amount of detail of the internal structure that the cell that can be seen because of the electron microscope. Now, what you need to do is all of those structures, see if you can find the functions of the labeled organelles and the structures, what function they perform in the cell, and describe how they are suited to this function. So this would be a good time for you to pause the video now and go to the textbook and do this task. Okay, so hopefully you found the textbook useful in helping you gather the information that you needed to be able to list and describe the functions of the different structures and organelles within the cell. And now, just to recap and to summarize what you should have done, here is a video from the organic chemistry tutor that looks at the organelles and their functions within a plant and an animal cell. So the video is a good summary. And you can just check whether you've got all the detail that is mentioned in this video.
In this video, we're going to do a basic review of the cell and the things inside of the cell, like the organelles, the tiny organs of the cell. So first we have the nucleus, which is right there. And the nucleus is basically the command and control center of the cell. It tells the cell what to do, how to grow, what proteins to make, what lipids to produce, and things like that. Now, in the nucleus, in this region, you have long strands of DNA known as chromatin. And the DNA is basically the instruction manual or the blueprint of the cell. It stores the genetic information of the cell. And so within the DNA, you have the instructions on how to make the proteins, how to make the individual organelles of the cell, and what they need to do. Now, inside of the nucleus, we have this ball right here, and that is the nucleolus. Now, the nucleolus, it creates ribosomal RNA to make ribosomes. And ribosomes, you can see these little spheres floating in the cytosol, and the ribosomes, they make proteins. Now, the ribosomes consist of ribosomal RNA and proteins, but they manufacture proteins. Now, the way the nucleus makes proteins is that it sends mRNA or messenger RNA, which carries the instructions to the ribosome to make the specific type of protein that's needed for the cell. Now, the nucleus is surrounded by a membrane known as the nuclear envelope. And on this membrane, you'll find these holes on it, which are known as nuclear pores, and they allow stuff to go into and out of the cell. So the messenger RNA comes out of the nucleus through those nuclear pores. Now let's talk about the ER, the endoplasmic reticulum. There are two types. The first one is the rough ER, which is all of this in that region, and the second is the smooth ER, which is right here. Now, what do you notice about these two? What is the difference between them? The rough ER contains ribosomes. The smooth ER does not contain any ribosomes. So the rough ER, it assists in the production of proteins because ribosomes, that's what they do, they make proteins. Because the smooth ER doesn't have any ribosomes, it does not make any proteins. However, it does produces lipids, cholesterol, and hormones. It also assists in detoxification, which means it breaks down toxins. And the way it does so is by making these molecules more water soluble so that they can be easily removed from the body or excreted through the urine. Now let's go back to the rough ER. So once the rough ER, by means of the ribosomes, once they make the proteins are enclosed in a vesicle, and those vesicles get transported to the Golgi body, which is right there. Now, the Golgi body, it receives the vesicles, and it modifies the proteins that are in the vesicles. And the way it does so is by adding lipids and carbs to the proteins, and it can also fold the protein. It can give it the proper shape because the function of a protein is dependent on its shape. So once it modifies and processes the proteins, it exports the proteins out of the cell. So here is a visual illustration of what happens to a protein in a cell. Now granted, my drawing is not the best. This is an educational video and not an artist or a how-to draw video. And so the protein, it begins at the rough ER because that's where the ribosomes are. And remember, ribosomes, their job is to make proteins. So the protein, it leaves the rough ER by means of a transport vesicle. And you can see the protein inside of this vesicle. And it goes to the Golgi body for processing and modification. So looking at the protein now, you can see that it has a different shape. At the same time, you could see different colors added to it. So this represents the lipids and the carbs added to the protein. So once the protein has been tagged with those extra molecules, it gets exported out of the cell. So it travels to the cell membrane where it leaves the cell to perform the function that it needs to do. Now the transport vesicle that surrounds the protein, it fuses with the cell membrane, and so it becomes part of the cell membrane, causing the membrane to expand, or basically causing the cell to grow. Now let's talk about some other organelles that you need to know. The next one is the mitochondria. Now the mitochondria has its own separate DNA. It's different from the DNA of the cell. And its job is to perform cellular respiration. In that process, it takes the energy stored in fats and carbohydrates and converts it to a molecule known as ATP, adenosine triphosphate. Next up, we have the lysosome. Now the lysosome, which is here, its purpose is to break down food. It contains digestive enzymes, and in white blood cells, it can also destroy pathogens. When a white blood cell engulfs a pathogen in a process known as phagocytosis, the lysosomes in those white blood cells, it can break down those pathogens or disease-causing agents into smaller stuff that can be used or recycled by the cell. Next up, we have the cytoplasm. The cytoplasm is basically the jelly-like fluid that is in the cell. So all of the organelles, they're dissolved in the cytoplasm. The cytoplasm contains solutes like salts, electrolytes, and other stuff like carbohydrates, lipids, and free-floating ribosomes and things like that. Now let's talk about the cytoskeleton. The function of the cytoskeleton is to maintain the shape of the cell. At the same time, it also provides structural support. Now the cytoskeleton consists of a network of 3 fibers. The first of which is the microtubules, the second is the microfilaments, and the third one, the intermediate filaments. Now how can we identify them in the cell? The microtubules is the largest of the 3 fibers. The microfilaments is the smallest of the 3 fibers. The intermediate filaments, in terms of size, it's somewhere in between the microtubules and the microfilaments. Now this picture really doesn't show it, but just know that the intermediate filaments, they're in between, and they provide mechanical support to the cell. Now the microtubules, they're made up of a protein called tubulin, and the microfilaments, they're made up of a protein called actin. The microtubules, their purpose is to provide structural support and transportation services to the cell. So vesicles, they can move from one part of the cell to another part by means of the microtubules. The microfilaments, they're involved in elongation and contraction, so they assist the cell to move. Next up, we have the centrioles. And here it is in the diagram. The centrioles are active during cell division. And during that time, they form something known as the mitotic spindle, which consists of microtubules. And what it does is during cell division, it pulls apart the chromosomes, thus allowing the cell to be split into two. The chromosomes are basically a condensed version of chromatin, so they form during cell division. Now, here are some other terms that you need to be familiar with: cilia and flagella. These also play a role in the movement of a cell. Now, cilia consist of tiny, short, hair-like structures, and notice that there's many of them. Flagella consist of long, whip-like structures, and there's only a few of them. And so the flagella can propel a cell forward from one location to another. Now, these two structures, they're made up of microtubules. So microtubules, they're very useful in the movement of a cell. So far, we've been talking about animal cells. But now let's talk about the plant cell, because even though they are similar, the plant cell has some characteristics that are not found in the animal cell. The first of which is the presence of a very large vacuole. This vacuole stores water and nutrients, and at the same time, it provides structural support to the cell by means of the hydrostatic pressure that it generates. Now, another organelle that's found in the plant cell, but not in an animal cell, is the chloroplast. Now, the chloroplast is green due to a pigment known as chlorophyll, which plays a role in photosynthesis. Photosynthesis is a process by which the plant takes sunlight, carbon dioxide, and water, and it produces glucose and oxygen gas. Another feature that's present in plant cells but not in animal cells is the presence of a cell wall. Now, both the animal cell and the plant cell, they both contain a cell membrane. The cell membrane consists of a phospholipid bilayer, and it has a property known as selective permeability, which means that it allows some things to enter while preventing the passage of other things from coming into the cell. And it has these protein channels. Now, small molecules like oxygen gas and water, they can diffuse right into the plasma membrane. But large molecules like glucose and other stuff, they have to go through a protein channel. Even ions have to travel through channels as well. And so that's the function of the cell membrane. It's basically like the gate of the cell. It allows some things to enter while blocking the passage of other stuff from going into the cell. So that's basically it for this video.